it's really close. Sorry, I know this is boring. I'm just waiting for everybody to trickle in. So, just chill for now. Yes, what's up?
wanted to let you know, I was trying this morning to sign up for Top Hat. I was having problems, so I did contact IT. So for some reason, that's what I reset, but I'm still having problems while I'm in.
So Top Hat is not Oklahoma State. So you're not going to access it with your OK account like that way. And you're also, IT has nothing to do with it.
Okay.
So,
Okay, well, that's good news.
so go back to like the course, like this course, and then go to the, I think it's in assignments, but also could be. One is set up differently, so I don't know always what it looks like for you guys.
Super techie person.
Yeah, so go ahead and just click. Okay, never mind. Yeah, go show by tab. Oh, okay. All right, and then click this. Should open in a new window. Maybe put that as different.
Nervous because I was like, "Oh, so funny, hide on it."
Because that link specifically should be the one that like links your account to Top Hat, and then you can access it after you have accessed it through that one link, like the normal way, like on Top Hat's website or through their app or whatever.
Okay.
So I mean that's what it's supposed to do to see if it opens up correctly.
If it doesn't, when you start, I can come back.
Yeah, we can try to figure it out.
Looks like my computer's gonna be running a little slow.
I understand. Yeah.
I'll let it
Just
work
let me,
on
yeah.
itself.
Let me know if it doesn't open and we'll try to figure out another way. Oh my gosh, autocorrect. Alrighty, alrighty. All right. So I guess we're right at time to start. I am Professor Yost. You can call me Lauren. You can call me Miss Yost. I don't care what, but I don't have a doctorate, so you don't have to call me doctor or anything. I just have a master's degree. Oh, we can talk another time about how that happened, but that's a whole story in and of itself. So, but yeah, most of my background, in case you guys haven't checked me out on campus, most of my background is industry. I've worked in industry, and I say industry, but what I really mean is labs in general. I did a lot of food testing, and I've done a lot of working in hospital labs. I've worked in just general, like mostly, it's all been in Integris, but Integris is like hospital lab areas, as well as their transplant lab. So their transplant immunology lab testing for people to see if they can, match to get organs and whatever like that. So I did that as well. So I have a background in actually using this work. And I've taught a few adjunct courses before that, but like most of my focus was in actually doing this work. So I have a slightly different perspective on it than professors who only teach and have never actually done the work. So that makes it a little bit more useful for most people. The fact that I've worked in an actual hospital lab and most of the stuff we're learning in microbiology applies to hospital labs and like what most of you being nursing students probably what you guys would be ordering. You need to understand what tests you're ordering and not just following the doctor's orders blindly. So it's my job to help you guys understand a little bit why you would be ordering those tests and what they mean. And hey, if you see something in your patient, what you can talk to the doctor or charge nurse about whenever you see something going on. Gives you a little bit more knowledge, capability when working with patients, right? So that's what microbiology is all about. And of course, a lot of it's going to be talking about bacteria. You don't really get to see them much except in your microscope, and they smell bad pretty much always. So there's that. And we'll talk about viruses and stuff like that. Most of the diseases and their symptoms and everything like that won't come until the end of the semester. But we'll be learning about how the cells work, how the viruses work, and how your body fights them off and everything like that up to that point. So that when we learn about the diseases, we're not just saying, hey, you have these symptoms, but we understand why you have the symptoms and everything. So it's hopefully going to break down some stuff that maybe you've had questions about, but never really thought to look into. So yeah. So that's the gist of the course. Every day I will be handing around a sign-in sheet. It's dry erase. So like when you pass it from person to person, just be sure you're not like erasing it accidentally. So it'll always be like that. Just be sure you sign next to your name. There are points associated with attending class. Okay, so you are like, if you want your points, if you want the credit for attending class, show up. If you don't care if you lose some points, that's on you, right? So we're adults and we can make those decisions. If you miss a class, and you have a verifiable excuse, so this is the thing, this is my one rule in my class. If you have a verifiable excuse, like a doctor's note for your kid, or you have an actual like receipt for having had have flat tire and some pictures to go with that, with dates associated with them, stuff like that. You can verify that these things happen to you. Then we can talk about like getting those points back. And I'm pretty lenient with it. However, last spring I had a student who she came into lab one day. It was the day before the exam. Some people have the nerve, man. So she she came in with like Coban wrap around her wrist, and she was like, "I was in the ER all last night. I can't take the exam today. I see, I've got this on my wrist. I can't take the exam." I was like, "Well, do you have a receipt or anything from the ER?" We all know. Well, maybe you guys haven't gone to the ER, but probably most of you know. When you go to the ER, they give you a crap load of paperwork to take home. It's like, hey, maybe you should stop smoking. Hey, maybe you should stop, drinking, even though you don't drink, and all this. And then also your, write up from your doctor and all that. So I'm like, do you have any of that you can show me that you got from having gone to the doctor? She's like, oh, I left it at home. So I'm like, okay, hey, we're not doing a whole lot in lab today. How about you go get that and you can bring it, and then you have your verifiable excuse to not take the exam. Because it's an exam, guys. It's like 10% of your grade in that one thing. And she's like, oh, it's not a big deal. And I'm like, no, really, it's cool. It's just the lab. You were here to see the first part of it. You got the gist of it. It's okay. I knew what was going on though, And she's like, okay, listen, no, I didn't have anything happen, right? I'm making this up. She finally broke down. She's like, I'm making this up. I just didn't want to, I'm not ready. I didn't study for the test. And I was like, if you guys come to me and you're like, hey, listen, I didn't study for the test, straight to my face, I'm more likely to hear you out than if you lie to me and try to get out of it that way, right? I have no respect for liars. So yeah, if you fuck up your patient, right, and you're at the hospital that you guys go work at or whatever, and you just try to cover it up with excuses, how quick do you think you're going to get fired? real fast. So don't lie to me. That's not, I'm here to help you guys have a better experience in your career. I'm literally here to help you. I'm not your enemy. I'm here to help you guys out. If you do have an issue where you couldn't stay for the test, I'm probably going to make you take the test. But if it really goes as badly as you're afraid it would go, like if you just bomb and everybody else did way better than you, then we can talk about potentially getting some other points to make that up. Hey, you did bad on the test. How about you do this other project and we can add that on or something like that, right? But if you lie to me, I'm not going to help you. That's pretty much how it's going to go. If you don't have respect for me, why should I have respect for you? So I'm just putting that, like, okay, signing in on the sign-in sheet is a different thing, right? But but you get what I'm saying. If you have a problem or something, come see me and we can talk about it. Be honest about your problems, and you'll be in a better place. That's my point. I'm pretty lenient for the most part. But yeah, this is where I'm going to be putting your attendance points. So if you come in late, and I'm not very specific about when you have to be here, I prefer people be on time because I'm going to start lecturing on time every day. But if you come in 15 minutes late, you just know that you've missed the first 15 minutes of lecture. Just be sure you sign in on the sign-in sheet. That doesn't really affect me, but let me know that you came in at least, okay? So be sure you sign in the sign-in sheet, even if you come in late, before you leave, okay? So that's on you guys. Make sure you take that, make sure you get that taken care of. I think attendance, which I call attendance and participation. And if you've like looked at all on the stuff of what this course is going to involve, then you probably know what I mean by participation. That is worth points. So it's 5% of your grade. It's not really a small amount. It could make a difference between your A and your B, or your B and your C, right? So show up, it's easy points. It counts if you show up to my class via Zoom. If you decide you can't do 10 a.m. here in person and you want to do 10 a.m. via Zoom, the Zoom link that I provided you guys in the announcements, the same Zoom link that we will always use the entire course. And usually I will repost it anyways because people still ask no matter what. But it's the same Zoom link. I'll always just be re-entering it. And you can join in live and participate. If you are joining in via Zoom, I still expect you to participate at the same level that you would if you were here as best you can, okay? And we'll go over what that means. But yeah, expect you to participate. And if for some reason you couldn't make it for whatever reason, I do record all the lectures. Or if you like to just review the lectures later on, I do record them and I will post them. In Unit 1, I just created like a, in-class lectures recording page. And I will be putting Monday, Wednesday, and Tuesday, Thursday lectures all on that one page. So you can find the one that you like, or if you want to watch Monday, Wednesdays, I don't know, whatever coach you wrote, whatever. But that'll be all on that one page for each chapter. So yeah, so there's those resources for you guys. So participation wise, so yeah, part of it is attendance. You have to actually be here. But if you don't participate regularly, so if one day you just like, I don't know what the answer is, and you're just not like into it or whatever, when I call people's names, which is part of what this class is about. If you don't participate one day, I don't care, it happens. We all have bad days and you didn't participate that one day and you didn't know or you weren't paying attention that day, fine. But if I routinely call on you and you never try to answer the question, I'm gonna remove your participation points for that unit. So if that whole unit you didn't participate, it's participation points too. So you might as well not be here if you're not gonna participate. Because if you're just going to sit there and just like zone out, why are you here? So, I mean, serious, like, why would you be here? So the whole idea is, if I tell you guys, you're sitting here listening to me blab on and on and on about whatever topic, bacteria and their cell walls and whatever, for an hour and 15 minutes. And if you ever experienced this with other teachers, especially if, let's say, and I love Dr. Shearer, but if you guys want to hear, an hour talk over how to do Morse code, he's the man to go to. So, but yeah, I love him as a person, but people talking and I talk fast and I talk like continuously pretty much. You zone out because it's early and we all have lives and we're thinking about other things, right? It happens. That's human, okay? My goal is halfway through the lecture and at the end of the lecture every day, I'm going to be calling on people for questions. It's 5 questions halfway through and another 5 questions the other half. And each halfway point is going to be questions about that previous half, okay? I will be drawing names. I have popsicle sticks. Today, when we get to it, I'll be calling your name. Some days I might just call in a group and we'll see how it goes. If, if I call alpha group, then anybody from alpha group can answer. But if I find out that like nobody is answering or just one person is always carrying the group, then we'll go back to regularly only doing names. But yeah, so be prepared. I want you guys to be aware that when we get to calling names, first of all, I expect everybody to stand up. Now, it sounds stupid and it sounds like kindergarten, but it works. It wakes your body up from sitting there zoning out. So you're not just sitting there vegging the whole way through. When we stand up to do the questions, it helps wake your body up and therefore your brain will be working a little bit more efficiently. So it's just to help us get our blood flowing a little bit better. It's only for the questions, okay? And I expect you not to use your notes for the questions either. So just recalling what you heard me saying. 90% of the time, you're going to get the answer wrong. That is okay, all right? How many of you guys would look at the person next to you getting the answer wrong and be like, oh my God, so stupid, right? Nobody cares, right? That person gets the answer wrong and you would probably be like, yeah, I didn't know it either. That's the more likely scenario, that this person guessed it wrong and most of the class probably wouldn't have known either. That's kind of the point though. So especially this first time, you guys are gonna be caught off guard a little bit, and that's okay. You'll get used to it as we go through the semester. It won't feel as weird or awkward. But when you get the answer wrong, try to guess based on what we've been talking about. I would recommend that. Like if you remember me talking about such and such thing, at least kind of be like, well, I remember it had to do with blah, blah, blah. I'm likely to give you the point. And I'll tell you that you were wrong. But, but I'll give you the point for trying. Okay, this is an effort thing more than it is anything. So if you try to put what we're learning into practice with the questions, then you're probably going to get the point. And it's a point for your group. It's not for you. Okay, so if you're part of Bravo group, then you got a point for your whole group. And I keep track of these points for the groups and post them on the front page of Canvas as we go on, so you can see where the groups are with their points. All right, if you look in, I don't know if I have it in the syllabus, I think it's in the actual assignment for the extra credit for this, because extra credit, okay? So extra credit points for the group. The group with the highest points at the end, and if there's more than one, they can all, if everybody gets the highest points, then everybody gets the highest of all the extra credit. But it would rank as like highest gets 100% of the points, next highest 90%, next highest 80%, no less ever than 60% of the points, okay? So that would be the absolute lowest. I don't anticipate that really being an issue. I'm sure there will be tied groups and stuff, but that's the idea. And if we have good participation, everybody's been trying the whole semester, the odds of me giving everybody full credit is pretty high, as long as you guys have been trying, okay? So keep that in mind. I'm really big on that sort of stuff. So in case you guys haven't gotten the big picture of this, this isn't about me just blabbing in front of you and it being entirely your responsibility to just figure it out afterwards. The idea is to get you guys more engaged. And that is going to help you remember the material better. Even if you don't get the right answer, I will be repeating the information to you with the right answer if you don't get it right. And I'll open it up. So if I, pick on so-and-so and, John Smith, and he gets it wrong, I will open it up to his group. Okay, well, John Smith's Bravo group. Anybody in Bravo group know the answer? Anybody in Bravo group can answer. And if they can't get it, then I'll open it up to everybody. And whatever group you are a part of, your group gets the points. And I'm not going to call on you later. Okay, so we'll do it that way and see how that goes. And I say see how this goes, but like we're going to keep doing this the whole semester, but we'll see how that ends up working for you guys as far as some of the questions I've had to sort of make a little bit more basic than what I originally put. That's okay. We'll do what we have to do to try to make it work for you guys. So there's that. The unit projects, the whole point we have the groups in general is for the unit projects. So units 1, 2, and 3 have a group project associated with them. And we have a day set aside for the group projects. And those group projects are essentially just going to be your group, Bravo group, let's say, creating a review over whatever the assigned topic was. So I think I don't remember what Bravo group's topic is, but off the top of my head, but alphas, I know it has to do with germ theory. Okay, so if alphas got germ theory, then the 4 of you are going to work together to make a review project. And it outlines some examples of what you can do on there. You don't have to adhere to that. If you think you have a better idea, just let me know ahead of time before you just submit it and surprise me. But things like making a poster, you make that digital or actual physical, old school, elementary school poster, or you could make a skit detailing how these things work together, or you could Create questions, but go over the questions with the class and what the right answers are and why. You can make a handout or an outline, but going over it with the class. But you're expected to know that material, master the material of that topic as a group, and then display it. And yes, you will be rating each other in the group. It'll be anonymous. Everybody in the group will be filling out a form, basically saying, okay, this person was 5 out of 5, or this person was 3 out of 5 for whatever reason. If you award somebody a 3 out of 5, let's hope that they're not just giving it to you because they don't like you, or you're not giving it to them because you don't like them. But if you get it a 3 out of 5 or less, you're required to justify why you're giving that person a 3 out of 5. Now, if it's just because they never showed up, then just write that. Okay, it doesn't have to be a novel about why you're giving them a 3. But I do want you to justify just so that I know that we're not just throwing somebody under the bus. Okay, but that'll be at the end of those projects. So you could think about it now. If you're in alpha group, you could be working with your other teammates to be working on that project for germ theory right now if you wanted to, or any of the other groups, whatever their topics are. But yeah, so it's basically you reviewing that topic to the class. So at least you'll have mastered that topic, hopefully, right? Whenever you get to the exam, hopefully you'll know that topic like the back of the end. And putting it in a different perspective other than me blabbing at it, because I can think about it the way that makes sense to me, but I don't always know how to put it in another words for you guys. So that's another way we can approach that, is seeing as it putting your peers, putting it in their own words. So hopefully you can understand those topics better. I also have, so there's that. So this is for you guys doing the work there. We will also have a review, like a proper review. And it'll usually be Kahoot led. So it's like questions, and I'll go over why the right answer is right and the wrong answers are wrong as we go through the Kahoot. And that'll be online on a Zoom at like, I don't know when exactly, because I have a late night Tuesday, Thursday class. So usually I've been having them like after that, but that's like late. So get ready for like 9 o'clock reviews for that. So it sucks, but that's when I can get it in for everybody all at once. So there's that, but we will have the reviews, and I do record everything, and anything that we would be doing, I will post on Canvas also for your review if you can't make it, because I know people have jobs and stuff, and kids and all that. So, and you can always join in via Zoom. If I'm recording it, I'm doing it live also, so you can always join in that way. So there's reviews that way. I also have the study sheet. I forgot to tell my Monday class about this. The study sheet is a sheet that has all the information that I'm going to test you over on the exam, that I could possibly test you over on the exam. I've narrowed it down from the information of everything that's in the text to these topics and what you need to know about these topics. That does not mean that that is the only thing you're going to see word for word on the exam. These are not questions that you're literally going to see on the test, right? If I tell you about how bacteria have cell walls made out of peptidoglycan, we'll get to that later what that means, but if I tell you they have cell walls made of peptidoglycan, the question might be, what are bacterial cell walls made out of? Easy enough. Or the question could be, a 55-year-old male shows up into your clinic complaining of, a fever and sore muscles and malaise and generally not feeling well. He also has, sore neck. His neck is really stiff and his head is sort of been throbbing, and he has sensitivity to light. And so maybe your doctor or your nurse is concerned about meningitis. So you get a CSF sample. And when you test that CSF sample before you look at the gram stain, you do detect peptidoglycan. What could be the cause of this individual's illness? And it would be as simple as bacteria, virus, protozoa, fungi. Now you guys know the answer right now because I just told you that bacteria have peptidoglycan cell walls. Nothing else has that, only bacteria do. So if you see that, then you would know automatically that this is a bacterial meningitis. And yes.
Does this class also translate to animal welfare as well?
Yes, there will be. I'll try to do my best to include the animal stuff. We do talk about some of the stuff directly, but the information itself, as far as like the bacteria and how they live and everything like that, of course, that doesn't matter if it's human or if it's animal. But that's a good question. I get that a lot from vet students. Of course, this class is geared more towards the nursing stuff, but we do talk about some of the animal infection as well. Yeah, that's a good one. Yeah, but you guys get the idea, right? It's more about application. So you would need to understand those topics well enough that you can apply it to a scenario like that. So be aware of that, all right? So that's the study sheet. It's the stuff that you need to understand well enough to answer questions like that about. It's a guide to help you study and understand.
That's in the study sheet.
Yes, that module, unit 1 at the very top, it's like study sheet and it says a guide to studying unit 1. I didn't want to call it study guide because a lot of people think that that means questions and answers that are literally going to be on the test. But these are concepts that I will test you over, and I won't test you outside of these concepts, but you do need to understand them. Okay. Okay. I think I'm good with that. There might be other stuff that we'll go over in the future, but we have a lot to cover today. So I want to just kind of move forward. Normally, and those of you guys who came in later, if you haven't signed the sign-in sheet, be sure that you do sign the sign-in sheet. That's going to be how you get your credit for attendance. Okay. All right. And we'll get into lab stuff when we get to the lab. I don't want to waste my time on that now. Normally, when we get into class, I will have a Kahoot up over the previous chapter. Obviously, we don't have a previous chapter, so we don't have that today. But we'll have 10 questions reviewing over whatever the previous chapter was. So next Thursday, this coming Thursday, when we meet again, we'll have a Kahoot over chapter 1 or 3 up, and then we'll go through that. Yes.
Are we kind of looking a little bit backwards instead of going 1, 2, 3, we go 1, 3, and 2?
Okay, so the reason we're going 1, 3, and 2 is because chapter 1 is going over like the basics of bacteria, right? Chapter 3 is talking about the tools that we use to study bacteria, and chapter 2 is talking about chemistry of bacteria. And to me, it made more sense to have 1 and 3 together. Oh. Yeah, and it's also really hard for me to summarize down the chemistry chapter because it's just a lot. A lot of people think that they did so well in chemistry and they understood it so well, but you probably did well because you were good at the formulas and you were good at filling out your little paperwork with your answers and your numbers. This is not, analytical class. This is going to be concept. So that sometimes can be difficult for some people. So that's why I have it set up that way anyways. But yeah, normally we would do that. This is the Kahoot page and I would click on it and we would do that. So next time we come in on Thursday, be sure you have your phones or your computers ready to do Kahoot. You just go to kahoot.it and then I'll have like a code or it'll be a QR code, either one that you can use to sign in, and then we'll go through the questions as a group so we can all see together how we're doing on it. Okay, cool. All right, so let's go ahead and get into the boring stuff. Why? So starting with chapter 1, like I said, it's intro to microbiology. What is microbiology? And I'll warn you guys, I am not Dr. Singh. I do not put a slide up and then talk forever about a slide. I will put a slide up and barely say anything that's on the slide and then move on. It's like a prompt for me that I'll talk about what you need to know. And you're welcome to go like reading through the slide. And if there's something on there that you're not getting, that I'm not saying, please let me know. Okay, if it's not clear to you. But the slides are there for like you need to be stay on track because yes, the slides are available on Canvas in that same unit. So if you unit in the modules, so you can find those in the modules as well. And the Kahoots are too, the ones that we're going to be doing before class. You can find those there too, so you can replay them for study purposes or whatever. All right, microbiology is the basically the study of anything organism that is too small to be seen with the naked eye. This would include things like bacteria, which we already know are tiny, or viruses, which we know are tiny, or anything else that would be microscopic. We can divide these into cellular versus acellular or non-cellular. Okay, cellular, literally, they're made of cells. They may be unicellular, single cell, or they may be multicellular, like we are. So we have bacteria, always single cell. Archaea, always single cell. Fungi, that's like molds, yeasts, and mushrooms, stuff like that. Protozoa, which we don't talk a whole lot about in society typically when we're saying things about protozoa, they usually don't get pointed out as protozoa, but I'm sure you guys have heard of some of them. You guys know about the brain-eating amoeba? Yeah, amoebas are protozoa. Okay, so they'll fall into that group. Some of those guys, a lot of those guys are going to be single-celled. And then we have the helminths. This is a word you haven't used, but again, another one of those that you do know what they are. These are the parasitic worms. So things like heartworms for dogs, and tapeworms, exactly, like you might get from pork, and all that stuff. So that's the helminths. They're the parasitic worms. Now, they obviously would be multicellular, right? You can see those guys, a lot of them, with your naked eye. So would we consider them microorganisms? Maybe not the adult forms for some of these, but because we often use their larvae or their eggs to identify them, and they are microscopic. That's why they fall into this category. Whenever you go to, if your dog has heartworms, I adopted a dog, two dogs actually, back in January that had heartworms, both of them. And it's a lot of work, basically, to take care of a dog when they have heartworms. You have to keep, like when they're being treated, you have to keep them in their kennel 24/7 pretty much. You can take them out, potty, but that's it. Yes. So parvo is a virus. Yeah, and it's actually one of the smallest viruses that there is. So that would be the non-cellular one. But yeah, so when you have heartworms, the way they're going to test you for the heartworms typically is with a blood sample. So they're not looking for the actual worms in the heart with that, right? They're looking for the larvae in the bloodstream to see if they're there. And you can't see those with the naked eye. So that's an example of elements that you might see kind of on day-to-day in human life in the United States anyways, right? Okay, so non-cellular organisms. These are guys that don't have true cells. What is a true cell? We would consider a cell to be something that is membrane bound, which we'll talk about what membranes are, no worries, but it has a membrane to contain everything. And everything, it can divide on its own. It doesn't rely on another cell to like cheat off of it in order to, steal its machinery to divide. It can do it on its own and exist on its own. That's kind of what a cell would be like and what we consider a living organism to be. Viruses, man, they walk this line. They can have a membrane, but they don't have anywhere near the amount of stuff that most other cells are going to have. And they don't always have a membrane. Parvo, for example, is so tiny, it doesn't have a membrane. It's just like a little bit of protein around some DNA, and that's it. And it's super tiny. It's one of the smallest viruses that we know of. So they can be anywhere from that tiny up to like some of the bigger ones like smallpox. It's actually a pretty big virus, almost the size of bacteria. So, but they're all viruses because they all have to infect a cell and steal its machinery in order to reproduce.
So like polio.
Yes, exactly like polio, yes. And then we have prions. Prions are misfolded proteins that just, once they misfold and they're not folded the way that they should, when we talk about chemistry, this will make more sense. I'll remind you guys about hydrophobic and hydrophilic stuff. But when they're misfolded, they are gonna like stick together instead of being normal in the cell. When they stick together, that can cause holes to form in the tissue where they are. And usually, typically in 99% of the time, we're talking about the nervous system, specifically the brain. So when they create holes in the brain, it makes the brain sort of look like a sponge with all the little tiny holes everywhere. It's called spongiform encephalopathy. And so what is the most common spongiform encephalopathy that we are probably aware of? Bovine spongiform encephalopathy, or mad cow disease. I know you've heard of that one probably. It's this very slow presenting disease. You can eat a burger that has a prion in it and not have any symptoms whatsoever for 30 years. But once you have symptoms, you usually die within a year.
What's it called again?
Encephalopathy, yes. You can probably, like, if you look up BSE, that's probably the easier way to like get into that, yeah.
I know in veterinary medicine, I know that. So brain.
Yes, enceph for brain.
Yeah.
Encephalo would be brain, and then pathy, like pathogenic disease. Yeah. Yeah. So you can think of it as being, we consider it a chronic or slow growing encephalitis, because eventually you'll have inflammation in the brain. The itis means the inflammation. So anyways, that's our intro to kind of what we're going to be talking about. Some of these guys fall into a category of cells that are so basic. All they are is just a membrane that surrounds the junk inside, but the junk inside isn't separated. So their DNA is just floating around, and everything that's going on in the cell is just floating around willy-nilly. Those guys are called eukaryotes. So A being without, karyo being a nucleus. We, however, we have a nucleus. I'm sure you guys are aware of that, right? We have all of our DNA contained within our nucleus. Our chromosomes are in our nucleus. It is a membrane-bound structure. And there's other membrane-bound structures that we have in our cells, like mitochondria, like endoplasmic reticulum. And maybe you haven't thought about these as being categorized that way, but that's what they are. They're individual membrane-bound units in the cell that have individual functions for the cell. Bacteria and archaea do not have that. They are eukaryotes, okay? They're cells, but they aren't compartmentalized. Eukaryotes, U means true. So true nucleus, karyo, those guys are the ones like us. We fall in that category. Okay. So if you are a species, you either are one or the other. You can't like have some that are mixed match. So yeah. So bacteria and archaea are our eukaryotes. Everybody else is going to be eukaryote. Okay. All right, obviously, if I'm talking to you guys about bacteria and archaea being, super basic, they just got this one membrane, everything just flowing around in there. That's nowhere near as complex as the little compartmentalized stuff that we see with eukaryotes like you and I. So which ones do you think came first in evolution, right? The bacteria, the simpler ones. So bacteria and archaea likely came first in evolution before the eukaryotes did. I'm going to tell you something cool about the eukaryotes. Those little membrane-bound structures that we have in our cells, they think that those came from like bacteria. Like there was a precursor cell back in the day, and it ate up a bacteria. And it found out that working with this bacteria and living with this bacteria, they were both getting a benefit, symbiosis, from that relationship. And so it stayed that way. There's a lot of evidence to support this, especially with mitochondria and with chloroplasts. They have their own DNA. Their DNA is more like bacteria DNA. We have so many chromosomes, they're all linear. You see like the X's, there's a line basically. And bacteria have circle chromosomes. Our mitochondria have circle chromosomes. Bacteria like gram-negative bacteria, we'll talk about those in chapter 4. Those guys have two membranes. So do our mitochondria and so do chloroplasts. They also have their own ribosomes. They're like bacterial ribosomes, not like our own. They divide on their own. Our cell, when it divides and does all this thing, it doesn't dictate when the mitochondria and chloroplasts are going to grow. They do that on their own. And they divide by binary fission, which is what bacteria do. Our cells don't do that. We do mitosis. So really cool stuff, right? So that's called the endosymbiotic theory of how eukaryotic cells came to be into existence. So pretty cool stuff. We can find bacteria pretty much everywhere and archaea everywhere. Archaea often live in the extreme environments. That's why we don't hear a whole lot about them. You can have some archaea that might live in your body, like there is a methanobrevibacter, something, I don't know what it's called exactly, but something that makes methane and is archaea, and it lives in your mouth. And if you have it, then you're way more prone to having like gum disease and gingivitis and stuff like that. So pretty interesting, or tooth decay in general and bad breath issues. But yeah, photosynthesis, bacteria, the original photosynthetic organisms will be the bacteria that just have chlorophyll. And that's how they became chloroplasts, right? They were able to do this first on their own, and then something ate them up and then used that for their benefit and became plants, right? It's pretty cool stuff. Most of the majority of our oxygen came from these guys. When they first started developing, they were using sun for their energy, and they started making oxygen as a byproduct, and they built our atmosphere how it is now. It didn't have oxygen before these guys came into existence. So they built our entire atmosphere. So when I ask you guys on a test, where does most of our atmosphere come from? It's not plants, it's microorganisms, bacteria and algae. They fall into the same category. Okay, so algae are protista, so like the protozoa are. So those guys are the ones that are most responsible for that. We already know that bacteria and fungi are going to be responsible for breaking down the dead things. I don't think we need to go over that, do we? That's why it smells bad. That's what rotting is about. Why does this not want me to see this slide? Okay. Yeah, okay. They live pretty much everywhere. We say microbes living in the Earth's crust. Think about microbes that are in our soil right now. Those microbes that are in our soil are very important to creating nitrogen sources that are usable by plants. Plants can't do that. Nitrogen from the environment, they can't just normally use it. They have to have bacteria to help process it first, and then they can use it. Same way that you guys can't just, go out in the sun and soak up energy, right? We have to have the plants turn it into energy first, and then we can eat them, or the animals that eat the plants. But it's the same thing in the end. So basically, these bacteria will come and change it into a usable form for the plants. With global warming, we're having a significant increase in temperatures in the tropics right now, to the point that the bacteria are having a hard time living in these soils. They're drying out, and it's just too hot. And so now places like Sri Lanka that are in the tropics like that, which are having these issues with global warming, their plants can't grow because the bacteria can't survive.
So would archaea thrive in that?
You could have archaea that could be living in that, but if they can't, turn that nitrogen into a usable source, it doesn't really benefit the plant or us. But yeah, they could potentially live in those places. Yeah, pretty much you can imagine any environment ever. something can live there, like in those deep sea vents and all that. All right. We know that we use microorganisms to make our bread. We use them to make penicillin, right? Mold led us to having penicillin and biotech, of course. Did you guys know that if you have diabetes and you take insulin, that your insulin came from genetically engineered bacteria to produce genetically engineered bacteria that produce human insulin? Prior to the advent of this sort of biotechnology, which is one of the first ones that came out that was used in medicine, prior to that, we had to have processed cows in order to get the insulin. And so it was way more expensive and less readily available. Like kill them and extract their insulin basically from their pancreas. I'm sure they used the other parts for other things, but yeah, they had to extract insulin from their pancreas. So, or I don't know how they did exactly back then, because it was like in the 1800s that they first discovered. It's like 1860 something when they first discovered that. But now the biotech stuff has let us like not have to kill cows all the time just for insulin. So that's made it cheaper. It's way easier, way cheaper to grow bacteria than it is cows, doing that. So, and before that, if you had diabetes, you just died. Legit. That's what happened. All right, so this is a term that we should all be familiar with. It's one you've heard, but now we're going to apply a definition to it, a pathogen. So if you hear that term, you're probably already thinking causes disease, right? Pathogens are literally microbes that cause disease. It's that simple, right? So not all microbes do cause disease. Do the ones that are in the soil helping the plants, do they cause disease? No, so they're not pathogens. Staph aureus can be a pathogen. Is it always a pathogen? No. Some people can have Staph aureus growing on their skin normally as part of their normal flora, about 4% of the population. But of course, if you're talking about MRSA and you can't treat it with antibiotics and it causes a skin infection, like cellulitis or something, then that can get out of control pretty quickly. And then it is a pathogen for sure, right? But if we're talking about something like the plague, Yersinia pestis causes the Black Plague. Is that a pathogen? Freaking always. It's never not a pathogen. Like, so.
Somewhere where it's melting down.
Yeah, new microbes that like are still alive. Like, yes, I heard about that. Yeah, and I think there were disease-causing microbes. I don't know if they can infect modern day man, but yeah, they'll probably be susceptible to the antibiotics though. So at least we'll have that. All righty. We talk about disease. Disease is any deviation from the norm. So if this is how you normally feel and you're deviating from that, that could be disease. We just mentioned diabetes. That's a disease, right? But is that caused by a pathogen? No. So not all diseases are. Heart disease and cancer typically are not caused by pathogens. Can be, but typically not. COVID is a pathogen. So that's a virus, right? And so that's a microbe now that we're talking about that does cause disease. That's just some examples. Not all diseases are caused by pathogens, but pathogens are microbes that do cause diseases. All right, so we have already kind of separated this out between eukarya, the true nucleus having guys, and the guys that don't have a true nucleus, which are archaea and bacteria only. Only archaea and bacteria do not have a true nucleus. So we can divide it out that way and then separate these guys out by their features, whether they are always single-celled or whether they are sometimes multicellular and all of that. And we can compare them by size as well. The bacteria cells are typically 10 to 20 times smaller than eukaryote cells. So already you've got a feature that you could go by. And also they don't have any organelles. Those like the mitochondria, like the endoplasmic reticulum, the nucleus, anything that's segmented off in the cell. If I were to give you a sample and put it on a slide and pop it under your microscope and ask you a question, is this a eukaryote or is this an eukaryote, like bacteria? Then you should be able to tell me by looking at the cell, it doesn't have any organelles in it, so this is going to be bacteria. I'm not going to give you archaea. That's not happening. It's weird. They're weird. They're not normal. So you don't see them a lot. So you'll mostly hear me talking about bacteria and eukaryotes in this course. And then, of course, the acellular microorganisms. The viruses that you cannot see them in a microscope like we have in our lab, you'd have to use an electron microscope that literally shoots electrons at a thing and then creates an image based on where the electrons bounce off of it. So that's how we can visualize viruses. You can even visualize entire, like individual atoms with that, which is pretty neat. No, we don't have those. It's really expensive. Our physics professor tried to petition to let us have one, but yeah, it's not happening. Prions are just proteins, so they're way even smaller. They're just a protein. So here are some images of some of these guys. I'm not going to talk about too much, but that upper left that I know you guys are all looking at, that is the tapeworm. Okay, that's the head of a tapeworm, and it'll embed that scolex head into your intestinal wall and just hang on for dear life and absorb all your nutrients. You guys know what the scientific method is. It's basically a means to answer a question in science, and your question we would call the hypothesis. So you've observed some things about life in general, and you say, I think this is what's causing this. That's your hypothesis. And then you're going to put it to test with your experiments and either prove or disprove that hypothesis. That's really all you need to know. I'm not going to go too much into detail over the scientific method. As long as you get the concept of a hypothesis, and we're either trying to prove or disprove it with our experiments. Now, if we prove our hypothesis with our experiments, that doesn't mean that it's instantly accepted as fact by the whole scientific community, right? We not only have to have repeat of that experiment to prove that you're always getting that result, we also have to do different experiments approaching it from other sides and proving that anything that would negate that is negative when we test that. And then you can maybe have a theory if you're lucky. And having a law is a whole other next level. So that's basically what this is talking about. So theories, it's a collection of statements that support the idea of a natural event or phenomenon explaining it. All right, that's all we need to know about it.
Wait, so
yes.
will it take quite some time for something to act, like Newton's law? So it takes an actual long time for it to actually come implemented?
Yeah, and it's not just so much a matter of time, so much as it will be like all these different experiments that have to align with the same answer as what it's proposing. And that's really what takes the time, is having all that happen to a degree that like the physics community is going to say, this is how this is, like the law of thermodynamics and stuff like that. So, all right, so Pasteur. Louis Pasteur disproved spontaneous generation. You probably see that statement up there and think like, cool, what is that then? So spontaneous generation is the thought back in like the 1800s and well before that disease, rot, anything like that, change in a matter like that, was spontaneous generation. So that means it came out of nowhere. If you put a steak out here and just leave it here for days and days and days, whether it's going to start rotting and smelling, or if we get maggots on it, right? We know that bacteria would be doing a lot of that, and the maggots would be from flies laying eggs on it. But they don't see the eggs when the flies land, right? How would they know that those were there? So they just thought, yeah, there might be flies and then there might be maggots, but how do you know that those are even related? So they thought all this stuff was just coming about, like when you reach a certain stage in your existence, that these things just happen spontaneously. They also believed in something called miasma. Miasma is like bad smells causing disease. So, walking in after your husband's been in the bathroom for a little while, yes, they would think that something like that could potentially cause disease. That's miasma. That's why whenever we talk about the plague and you see the plague doctor masks, that's the reason they're shaped the way that they are, because those beaks are how we see them as beaks. On a mask, they shove herbs and flowers in there so it smelled nice, because they thought if they smelled nice things, they can't get sick, which is nuts. But they didn't know that. But yeah, miasma. So spontaneous generation and miasma was the explanation for everything back then. Pasteur thought that was BS. So what he did is he took these flasks with these swan necks, boiled some media in it to remove anything that could be living in there. And then he cut off the neck of one of them so that the air could get in there more easily. So the one that he cut off the neck on grew stuff in it. And the one that had the long neck that was harder for microbes to get up from airflow and stuff like that didn't grow anything. And so he said, if this is spontaneous generation, that one with the long neck should have grown something. It shouldn't have mattered that it had the long neck, because it already could have spontaneously generated here. Why didn't it? It didn't. So he said, load of honky. And what's really going on is there's like stuff that's so tiny that we can't see it that's causing this stuff, microbes, or they call it germs. So this is, yes, they probably did call me heretic. But yeah, so Pasteur came up with this idea of germ theory. So that's what his concept was, germ theory, that if you had the introduction of growth into something, whether it's a person getting sick or media like what we just saw, germs had to do it, microscopic guys. Robert Koch was a guy that came up with steps to identify specific diseases based on this. And then Holmes and Semmelweis especially, they noticed that women who gave birth at home were getting less sick than the ones who gave birth in the hospital. That's because the guys that were in the hospital, the doctors in the hospital, were going and doing autopsies, and they were not washing their hands before coming and dealing with these women who were giving birth. And so they were dying. And so Semmelweis said, now we wash our hands now. How about we don't do it like this? And then we also have Lister, Joseph Lister. Yes, Listerine, okay? He was the first to utilize hand washing and misting operating rooms with aseptic chemicals. The chemical that he used was carbolic acid, it's also known as phenol. It's very harmful to people, so we don't really use it a lot today. And originally, Listerine was phenol based. It isn't anymore because it's harmful, but that's where Listerine came from.
Wait, they didn't swallow that though, right?
No. I mean, I don't know, back then, like back in these days in the 1800s, the stuff that they were, consuming, like lead paint and all of this, and like even the hats could make you sick. Like there's just chemicals in like fabrics and stuff. So they don't know, but at least for now, the concepts have been spread down into modern medicine for what we're thankful for actually now, right? We're glad that they clean your operating rooms now. All right, and then Pasteur, coming back to him with his germ theory, he also invented pasteurization. If you drink milk here in the United States, typically it's going to be pasteurized. Buy at the store, it's pasteurized.
Isn't that why some cheese isn't allowed?
Yes, some cheese isn't allowed in the United States, and you can have unpasteurized milk if you like go buy it from the Amish and stuff like that, but I don't recommend it. People say it's better for you because it's got all these bacteria that are so good for you, but are they bacteria that are really so good for you, or are they just bacteria? Is that you can't put them in the same category just because.
Wait, has anyone tested that?
I mean, people get sick from unpasteurized milk all the time, but it doesn't mean you always will. So because some people don't, they think that means that, those are the good milk or whatever. I don't know. But pasteurization involves like heating and cooling liquids so you don't damage the property of the liquid, but you are killing microbes by doing it. So that's pasteurization. And he also showed human disease could arise from infection. He took those flasks even farther and showed that people were getting sick from a similar principle. Robert Koch, again, he had steps that he could use to prove the cause of diseases that fell in line with germ theory. Now, we can classify and name our organisms. Just that helps us basically identify them later. So like if you get a sample and you're not sure what it is, and you're doing all these tests to figure it out, the fact that we have them categorized based on features helps us break it down into what it could be as we're going through. So if you do this test and you do gram stain, which should be the first test you start off with bacteria, gram stain, if it's positive or negative, will let you help go down the line of, hey, you don't normally test this stuff for gram positives. This is a gram negative, so why would you be doing the gram positive tests on it, right? That helps too. Let alone following and just doing a bunch of tests and then getting your answer. So that's the point of categorizing them. Binomial nomenclature is something I am going to test you on. It's going to be the most important. So first, on the first exam, I will have examples that you can pick from. When we have it typed out, it should be italicized, number one. So italicized, first letter should be capitalized. The first letter of the second word, the species part, should be lowercase. So one way you can remember it, you remember seeing about E. coli, right? You think of E. coli, it's usually capital E and a little c for the coli. Just remember about that and you've got it figured out. Yeah, E dot is, yes, but that's just the abbreviation, right? So the whole name of it is Escherichia coli. But as long as you can remember that concept of how it's written with the capital and then the lower c, italicize it when it's typed, okay? Because I'll have it on the test and I'll have to pick the right one. If you were writing it by hand, you would underline it. Technically, I'm never going to test you on that though. All right. Well, like if I were having you guys write answer tests, because I don't have short answer tests, they're all multiple choice. Because I don't want to read people's handwriting. but yeah, if I were to have you write it out, then I would expect you guys to underline it then. But not everybody's sure, but that's also up for me to determine, right? Not you guys. Abbreviations, like we said, E. coli, S. aureus. I mean, I don't know how else to break it down more than that. Taxonomy. I know you guys learned this in elementary school, probably. You remember kingdom, phylum, and all that. You got to know it again, and I am going to test you on this one. So get whatever, mnemonic that you need to help remember King Philip, whatever. And remember it so you can write it down on a list. And then when I ask you which one comes after phylum, then you can choose the correct one. Oh yeah, it's like a pyramid, but you guys just have to know it as a list. And this is pretty much the only chapter I'm ever going to test you about with it. But yes, here it is. We start off with domain now, and all domain is, just so you know, it's whether you are eukaryotes or you're not. So eukaryotes versus bacteria and archaea. So there's 3 groups that fall into the domain: eukaryotes, bacteria, and archaea. Remember, bacteria and archaea, they don't have true nuclei. Okay. I'm not going to go into too much detail on the differences between those, honestly, because I just don't care enough. But yeah, so that's domain. That's all domain is. I'm not going to break down any of the other ones. You just have to remember them in order. All right. Kingdom, phylum, class, order, family, genus, species. I don't know if King Philip could, I don't know, offer family good spirits. I don't know. Something, whatever works for you. And then you can remember that saying and then write those letters down. And now you got it. If I ask you which one comes before order and I give you family, genus, species, and phylum, then you can pick phylum. And that's from most general to most specific. Phylogeny is literally just looking at the genetics of organisms and then dividing them based on their genetic differences. Typically, when we look at the genetic differences of organisms, we're looking at their RNA, basically of their ribosomes. The ribosomes are the machines that make your protein. We'll talk about that in chapter 6. We're not there, but they are something that every cell has. Every true cell has ribosomes. And because they have ribosomes, we can compare the difference in the genes that are coding for those ribosomes. They have ribosomes, yes. So bacteria, archaea, eukaryotes all have ribosomes. Viruses don't, and prions don't, because there's protein. But yeah, so they don't. But all the other, anything that is made of a cell at all, they will have ribosomes. And we can compare the similarities between the genes coding for the ribosomes and see how related they are. That's basically phylogeny, what they do. Yeah, I don't know what else to say. Okay, this is how we're going to do it. Remember how I told you guys we're going to stand up, right? So everybody go ahead and stand up, humor me here. And I'm going to start calling on you guys for these questions. I've already got my first victim. All right, thank you. All right, first one. What is a pathogen? I've got Rachel L. Just give me your best guess. So good guess. Anything that causes disease, any organism that causes disease. So she didn't get it exactly right, but she's on the right page, right? So she gets the point. So don't freak out too much. Remember, this isn't a test, really. It's just to see if you guys can remember what I was talking about. So germ theory. Let's see. What is germ theory? This is not got anything written on it. I have some blank ones in here since you guys don't have a full class. All right. Krista S. Germ theory. You didn't used to be Krista S, I'm assuming? Okay, so probably Krista S isn't here. That's okay. I got another one. Jennifer S. Also not here? We have some people not here. Huh? That's probably you. Yeah. Okay. So germ theory, what do you think it is? Yeah, so yeah, that's the general gist of it. Germ theory is that concept that if you've got disease or rot, it had to come from a germ, right? So that's all germ theory is saying. So exactly, so how that, whatever it is, it could be a virus, had to be what caused the disease. All right, can you name one non-cellular pathogen? Carissa E. Yeah, that's fine. That works. That works for me. Yes. So that works. And if we were to get more specific, we could even say something like COVID or the flu if we were being specific. And I don't need you to. That's fine. All right, next one. One level of taxonomic classification, Carissa F. Yep, that'll work. That's fine. We've got the kingdom, phylum, class, order, family, genus, species. I literally have to say them all at once if I say any of them. So that's why I do it that way. But yes, that works. And then we've got, what is the name of the scientist who was the father of germ theory? What was his name? And we have Alicia W. Yes, that's faster. Exactly right. So we have 2 points for Foxtrot, 1 for Delta, 1 for Bravo, 1 for Echo. So good job so far. We've still got 5 more questions. You guys can sit down now. I'm going to start chapter 3. We're going to try to knock through it really quick because we don't have a ton of time left. We've got the 5 I's. This is probably the bread and butter of trying to figure out what a microbe is when we're trying to identify, an unknown of some kind. So the 5 I's. Number 1 is inoculation. Number 2 is incubation. Number 3 is isolation. Number 4 is inspection. And then number 5 is identification. So we're going to go through each of these. So inoculation is just producing a culture. And a culture is when we're growing microbes. That's all that means. So yeah, but with microbes, yeah, yes. So we have a medium, which is supposed to be the single of media. Now, I'm not gonna lie, you're really only ever gonna hear me say media. I'll be like, oh, this is our media for the day. Or I never say medium, just FYI. So media is our nutrient-rich stuff that we'll be growing our microbe on or in. Inoculation, that's a term referring to introducing those microbes to that media. Okay. And then sterile means it's free of all life. So you want your media to be sterile. So you want it contaminated with something that you're not studying, right? There's different kinds of media, and I'm not going to really go over it in this slide because it's going to go over it in the next. Liquid media is just going to be like broths. If it's liquid, I don't know. If it's liquid, it's liquid. If it's semi-solid, here we're talking about this is clot-like or gelatin almost. So think of like a soft jello. That's semi-solid. Okay. Then we have solid media. Solid media is given its solid aspect from agar. And I know you've heard of that one. So agar is actually a polysaccharide from algae. That's what I'm more likely to ask you guys on the lab stuff, because we'll talk about this stuff in lab, obviously. But that, you add it to media in certain concentrations can make it more or less solid. So anywhere from 1 to 5%. And it's not hard like this. It's like just a more firm jello, really. Think like gummy bear, like it takes pressure to break the surface. All right, we have defined media where we know everything that's in it. So this just means we know the exact chemical composition. We know that there's, 10% of sodium chloride and whatever in it. Now, why would you not know the exact composition? Because you have complex media. So this will be containing extracts from animals. We have a lot more of this than you might think. Yeast extract is a very common additive to our general growth medias. And so it's just giving them the general amount of protein and other nutrients. We don't know exactly the concentration of each one in there, right? Because we didn't put it in there hand by hand. So if we know the exact concentration, we put it in there hand by hand, that's defined. If we're using blood or an extract or something that can't be defined, but we know that it's good for this media, then that's complex. All right, there's general purpose media for just growing pretty much anything. That's all it really is. And then there's enriched media. Enriched media is where we add a special factor called a growth factor to it that organism cannot make for themselves. So they have to have it added in. So again, that factor is called a growth factor. And if you are an organism that needs that, then you are fastidious. So some organisms can't survive without certain things being added to the media. Hate to break it to you, but you are fastidious, right? So you guys know what happens to you if you don't get enough vitamin C. You get scurvy and you die eventually. But that's because no matter what I feed you, if it doesn't have vitamin C in it, you get scurvy because your body can't make its own vitamin C. Same thing with B12. I'm sure some of the women in here know what I'm talking about. Some women have a hard time absorbing B12 from their food. And so they have to get shots or take supplements or something like that in order to maintain their B12 levels. Even then, you still would have to get it if you're healthy, otherwise, from your food. You can't make it yourself. So vitamins a lot of times fall into that category. Okay. All right, so that's enriched media. That's what a growth factor is. Then we have selective media and differential media. So we look at this. Usually I have a little tablet I can use to draw, but they don't have it here anymore. Look at this side. This would be selective media. I think this says it down here in case you are unsure. That's the selective media. We take a sample. Let's say I swab the back of my throat. Then I put it in a Teflon swab, whatever. is there more than one bacteria on there? Probably, right? There's a ton of bacteria that grow in your body on it, whatever. So if you put it all over your plate and look at it to grow, you're just gonna have a mixture of all these different kinds of bacteria on there, right? But if you're pretty sure that your patient had strep throat and you wanted to select for strep bacteria, then you might use a media that really favors the growth of strep as opposed to any other kinds of bacteria. And that would be selective media. So you plate it all on there on the regular media, but if you put it on the selective media, only the things of interest are going to grow. Selective media selects for the thing you're interested in. Literally only that will be growing. All right, the other one is differential media. And that one's on the right. In differential media, we would take that throat swab and put it on a special plate. And then some of those colonies might be white, some of them might be blue, some of them might be red. And based on what color they change means that they were able to break down something in the media and turn it to this byproduct that causes the color change. So almost always differential media is going to have a color change involved, and it allows you to see the difference. That's what it is. So selective lets you select for the one you're interested in. Differential lets you tell the difference, but it's not anything can grow, technically, right? Okay. So selective, only certain things can grow. Differential, we're really only looking at color changes. And that's usually due to metabolic byproducts, something that we'll be doing in lab. At the top, this is called it's called triple sugar iron agar. And at the top, we have that pinkish color, right? In the middle, we have that black on the bottom. And on the right, we have the yellow. It's actually been lifted up inside of the glass because there's some gas production and all that. All of that, those are the same media. It's just after they've been grown with bacteria, two different kinds. Those things tell you stuff about that bacteria. And it's not just a matter of did it make black or did it turn yellow and make gas. It's like, what does that mean if it turns yellow and make gas? In this case, it means it ferments glucose. I don't even know that yet, but it tells you stuff about their metabolism. So that's what differential media can do for us. How they react to the media. All right, blood agar is a good example of a differential media. We're looking for hemolysin. It's literally just agar with some blood mixed in, sheep's blood mixed into it. And if your bacteria can break down the red blood cells in the media or not. So if they can break it down completely, then you'll see a clearing around where the growth is happening. If they can only break them down partially, then they'll have a greenish tinge from breaking down the cells but releasing the iron, but not completely. And then gamma hemolysis means you grew, but you didn't see these major changes. Oh, geez. All right, so up here we can see Enterococcus, it grew, but didn't have any major changes. Look at the alpha over here is the alpha hemolysis. This is a special kind of strep called strep sanguinis, I believe, or sanguineus or something like that. Anyways, it clearly is greenish, but it's not cleared the media. Like what you see with strep pyogenes, you can very clearly see that there's a clearing around the growth there. And then when you compare our patient sample, if you were just comparing it to these, you would say that it is most likely going to be strep pyogenes.
Are there basically multiple variations on the whole thing? So like with strep, there's multiple variations of it?
Yes. So like, for example, so strep, obviously they had strep sanguini or whatever that is. That one has to do with, it's the normal one found in your normal flora. But anyways, staph, right? Staph aureus, we know that that's going to be a disease-causing one, but there's also Staphylococcus epidermidis, which I'm sure you can imagine is just normal growth on your skin. So yeah, so there's a whole bunch of different kinds of species that can fall into each of those. Question. So we also can have media that can be both selective and differential at the same time. I'm sure you guys knew I was going to go in this direction. So, so this media starts out red, and you can see it's turned yellow in some cases. Whether it grows on there or not is going to be determined by its ability to handle salt in its environment. Things that grow on your skin are usually pretty resistant to salt. So this is usually used for Staphylococcus species. Things that aren't adapted to the skin won't be able to grow on it. Now we've got things that can growing on it, but how do we tell the difference between the things that did grow? And so that's where the color change came from. If you were to streak a sample on there and it stayed red, it grew, but it stayed red, probably going to be Staphylococcus epidermidis. But if it turns yellow, then it's probably Staphylococcus aureus. And that is because it can break down the mannitol sugar into an acid. That causes a pH change, and that's what causes the color change in media. We'll go over the details of that. You don't have to know that right now for the test, but that just gives you an idea of an example, okay? We'll go over the details of that, and you will have to know it for the lab, but that's the second half of the lab, y'all. That's not right now. So, but it is a good example. You can see how it could be useful. Okay, a colony is just, you look at a plate of bacteria and you see those little dots on it. That's a colony. Okay. It's a discrete mound of cells that originated from one single cell. So each mound came from one single cell just dividing up, creating its little mound. So if you have little mounds, little colonies touching each other, and they're all different colors, you can't really say they're pure, right? But if you had them individual and they weren't touching each other, you could pick up one colony with your little loop and use that to create a new pure culture. One way you can get those individual colonies is the streak plate method. I'm going to talk about this more a lot in lab, and you have to know for this as well. But basically, you're just putting the sample down on there and turning it and then spreading it again, turning it and spreading it again, and then turning it and spreading it one more time. And you're sterilizing your little instrument every time in between. Why? Because it's going to basically dilute out all the sample that you put on that plate. Okay? The idea is to dilute it out so we can get individual colonies. You can see that they got individual colonies from that mix by doing exactly that. Now they can pick up one of these and create a new culture with just one. That's the streak plate method. That's probably the most important one for isolation. So now we can create that pure culture. Our pure culture will be anexic, meaning it's free of other living things, except for the thing we're studying. That would be also a subculture. If we had this plate, this is already a culture, right? We cultured it on this, but with the streak plate method. So if we pick off one of these colonies, put it into a broth, that's a subculture. Since we're taking a culture from another culture. Okay. All right. We obviously can have mixed cultures. So if I swab my throat and I do put it on that plate, and it is a mixture of different kinds of stuff, that's a mixed culture. It started out that way. It was mixed to begin with. But a contaminated culture is a different thing. If I pick that single colony that we saw on there and create a growth with it, and then a couple weeks later I'm still working with it, and then I streak it, and there's a couple different things on it, then it's probably contaminated because it wasn't there earlier. So, all sorts of reasons how you could get contaminated, but remember that those are different. Mixed cultures, they started out that way from your sample. Contaminated, it got introduced later on. There's all sorts of different ways we can look at different kinds of microbes. Most of this stuff is going to be in a lot more detail as we go through the course. So I'm not going to go into too much different detail about this right now. But when we talk about biochemical tests, we're talking about looking at things like we did with the color changing media, looking at whether your organism can break down mannitol or if it can survive on salt and stuff like that. It tells you stuff about how that organism can survive. That's a biochemical test. It's telling you about their metabolism and stuff like that. You can also look at their genes, of course. You can do PCR on a lot of stuff nowadays. This is just looking at the different size of things. I'm not going to go over it right now, but you can see that prions are just protein down here. Here we have poliovirus, super tiny. Here we have HIV, right? And then we have E. coli all the way up here. And we have red blood cells. This guy that's above it is Giardia lamblia. He's a protozoan that causes horrible diarrhea. It's like you would have like a month or two of diarrhea, and it causes, they call it greasy malodorous stools. And it's from, if you go camping or whatever, and you think you're getting nice clear water from your stream, and then you go to drink it and whatever, but there's a dead animal upstream. That's Giardia typically. That's going to be causing that. So it's pretty awful. You can boil it and it'll be safe. No, it's not truly, I mean, it could be if you're like really not getting enough fluids or something, but yeah, typically it's not life-threatening. It's just going to last forever and you're going to be really uncomfortable. So how do we look at our microbes? Through magnification, obviously. Glass that has curvature to it allows us to magnify things, make them look bigger. Refraction is a term that is talking about the bending of light going through different materials. It's really important to understand that when we're talking about our microscopy later on. So we're enlarging our images and we're using light to basically transmit our image through a microscope. And we can determine the amount of magnification based on how many magnifying lenses we're going through. Now, our microscopes have two that you'll go through. We have a whole bunch down at the bottom. You can see even down here, it's just a good example. You turn this wheel and you have a whole bunch of these objective lenses hanging off, and you can switch between the different ones. And depending on which one you're looking through, it has more or less magnification, right? So it's our first lens that we'll go through. The second lens that it goes through is the one that you're looking at with your eyes. So you might be at your 4X, that's the lowest one, that's what they have it on here. And then so that's going to provide you just at that level 4 times what you're looking at. So it's 4 times as big as nature, okay? After it goes through some mirrors and then back through your eye piece, that's another 10X of magnification. So how do we calculate the whole magnification of it? From the 4X and then the 10X, well, you multiply them together. So it's just 40X overall, okay? It's always gonna be like that, and our microscope is always gonna be 10X on our eyepiece. So if I ever ask you about our microscope magnification, and I have it set on a 10X objective lens, and I ask you what the total magnification of the microscope is, it's always gonna be 10 times the 10 of the eyepiece, so 100. So you just multiply. It's always multiplying by 10, really. Whatever your objective lens is, times 10. Okay. It's not too bad. And we'll go over that again. We'll go over the parts as well in the lab. It just makes more sense to go over it in there than it does here. So we're concerned about magnification, resolution, which is our clarity, and then contrast, that's the definition. It's the difference between seeing something that's light gray on a dark gray background versus like white on a black background. So we're going to talk about these. Oh yeah, the ocular lens versus the objective lens. Your sample is coming through, sitting on a stage, right, on your little mic, on your slide. Light comes through it from the bottom from your condenser and shines in and hits the first lens, right? That's the very first lens we're hitting, and that objective lens, whatever one that you're rotated onto, that is the first image you're creating with a lens. So that's called the real image, okay? It's gonna go through some crazy nut job stuff inside of the microscope with, you know, mirrors and stuff. And then it's gonna go through another lens at your eyes, okay? So we're manipulating the image that time again. So what we had the real image being now manipulated again, that's the virtual image that you're actually looking at through your eye, because we've gone through mirrors and more magnification, okay? The one closest to the specimen, think of that as the real one. Cool, all right, we did this already. Oil immersion is basically, we take oil, it has a more similar property to glass than air does. So when we add the oil on there, it keeps the light rays in place. 90% of what we're doing in the laboratory on microscopes will be done on the 100X lens, which I can almost guarantee you many of you have used. And we'll be using oil immersion. So you're gonna get real friendly with some oil immersion. And I often have students who have taken physiology and they're like, oh, we use microscopes. I know how to use microscopes. Get ready. You don't know how to use microscopes. I can promise you. But that's what I'm there for, right? So I'll be there to walk you through all this and help you find your images so you can feel confident when you're looking through it, and we'll get there to where you'll feel pretty good about it as we go through. So, but yeah, the oil helps keep all of those light rays that give you all those details in place. So reduces refraction or light scattering. Oil reduces light scattering. That gives us also a little bit more contrast. And of course we can stain our samples to get contrast as well. I promise them to be done. Wet mounts are something that you can do where literally you take your broth and you put it on a slide and then you look at it right away. Like you do nothing else to it. You're just taking like the liquid and looking at it, but that can damage some really large cells and it can dry out. That's a big issue that we will have in our lab when we do our wet mounts. It's good for looking at like motility. So actually looking at your cells swimming around, which we're going to do, it's really good for that. But most of the time we'll be doing fixed stained smears, taking a sample, spreading it out on a slide, drying it out, and then staining it and looking at it. Most of the time we'll also be heat fixing. Heat fixing allows us to kill the cells and adhere the specimen to the slide. So when we're doing all of our rinses, it stays put. And this will also preserve cellular components for what it's worth. Okay. We will be doing a lot of this in lab as well, heat fixing. I really don't need you to know about basic versus acidic, but I do want you to focus on positive versus negative stains. We know that cells in general have a negative charge on the outside because of phospholipids on our membranes. And if you don't remember that, it's okay. Next chapter will explain that. But lipids are not, yes, they're kind of like fats, but yeah, hydrophobic, right? So we have our phospho groups are charged negatively, and so they make the heads that will kind of hide the tails on the inside of our lipid bilayers. Anyways, so the negative charge, negative repels negative, just like a South Pole magnet repels a South Pole magnet. So if you have a negative stain, it's going to repel the negative surface of the cell and you stain the background. If you have a positive stain, it's going to be attracted to the negative surface of the cell and stain the actual cell itself. Okay. Cool. This is an example of that. Because you create a background with your negative stain, this is a good example of it. And we'll be doing that with our capsule stains. So it's just showing simple stain that's using one dye to look at something. And these are some examples of some differential stains. We're going to get into more detail in chapter 4 and in lab for sure. Like our first labs are all this. So yeah, I'm not going to go into detail of them right now. Spore capsule flagella, looking at motility. Is what vectors. We're not going to do this because we're kind of pressed for time right now. So we're just going to go. But I will see you guys on Thursday. We'll have more time then, so I don't have to introduce everything. Yeah, that will be next week. Yes.
So you don't have to
Yes, typically it'll be lecture and then immediately lab will follow. But now I have to grow up all the cultures and get all those ready and everything like that. So I haven't got those growing yet. So we'll have them on the second floor. Oh, like for McGraw-Hill? Yes, it'll still count. And if I call your name on Zoom, then I would expect you to participate at least through like typing through the chat or something. If that makes sense. Okay.
Out of an hour, so
I understand.
I don't have like.
Yeah.
Thank you.
Yeah. That's a good question. I wonder where it loaded off to. Yes, it's just on that top hat. Okay, yeah. Thank you. Pretty easily, yeah. Okay, forgive you.
I just have a question about so when you put that aside,
Yeah, that'll be unit 4. And we'll start with typically what we'll do is like if you pick the disease like skin, then you'll start, presenting on the skin day or whatever. So we have chapters that are like each system of the body. And so you'll present on whatever day your system is.
and then you change how we're going to do that.
Yeah. Yeah, that's like a review. They will be worth points, those technically. So I know like before we were taking pictures of them and all that. So this is just like just for review. And you are welcome to use them for study purposes, but only not when we do for
I just tried to double check the things that I tried to take this test, but things are different.
Yeah, it is different. So you're good. Yeah, yeah, that'll be it. So the only thing you really need to worry about for points for even the lecture will be make sure you sign a sign sheet.
Thank you. I have more questions. Right?
Yeah, you can always email me. Yeah, so.
I watched the I think it was what 4. Just wanted to make sure. Well, thank you.
Yeah. So, attendance, as far as like, so there are points associated with attendance. But you can attend the class in person or you can attend on Zoom, so whatever works for you. But just be sure that you're aware of the fact that we have like participation, like what would have been this. I call on people's names and then ask questions and then they earn points for your group. So that way, people engaged. It's not about being right or wrong so much, but yeah. So if you do decide to do it by Zoom, just be aware you'll still expect people to answer if I call on you. I just pick random names out of the thing, but you can just type it and not. They are. I'll have to assign you to one, which is your problem. You have to open it, so I do it there and I'll try to email you when I do add you. It's adding a little bit late, but I think that's it for lecture. Yeah, and then otherwise lab starts next week. Yeah. Yeah. Thanks.
Our labs, what is the highest height?
Typically we don't go above 1. There might be a few organisms that technically are 2, but they're not typically at the settings that we're opening. You're welcome to bring your own gloves though if you want to. It's allowed. It's just we don't have the money to provide gloves to everybody. So yeah, that's what it's really about. Yeah, we used to have more twos, but I think it really was about like this. Now it's mostly ones. I'm used to that, right? I guess I was trained is like to always have the gloves, but now I have to like tell myself like, yeah, it's not really good for you. Only wash your hands afterwards. Yeah. Yes. Some groups will have 3, so you might, depending on what group you're in, your echo. I might be adding another person in because people are still adding the class. So that's.
I personally prefer mortality.
I mean, I don't really have any real recommendation for that other than like you're welcome to reach out to them now if you want to. You should be able to like put on their names and put it either, Maybe it's also possible you can try that. See if you guys decide to start doing it. And you can just send it to your entire group. Yeah, there you go. Okay, cool. So if you did have somebody new added onto there, it would just populate in that. So next time you email it, it will go back up to everybody. So yeah, you should be okay.
I'm not much of people person, but I'm trying to figure out like what to say, I guess.
Yeah, I mean, I would just, approach it as like, hey, we're working on this. Do you know what your topic is? I don't know what your topic is. It's in the actual group assignment, like so the unit 1 group assignments. And then you can sort it by yeah, and then that's what you guys I would just if you guys, anybody want to get started on their project or or like what you guys want to do, like even ask them like what they want to do or what kind of. I think that's a good place to start.
Is that what that is about? Like with like mass
So it'll be like this.
Yeah.
So it'll be dry erase, it'll be like a whole worksheet, and you can fill it out on the worksheet and then take a picture and upload that. I mean, they're on there. If you go to the lab module, the two modules, then lab documents and worksheets.
That's what I just prefer doing. It's a lot smoother for me, except in the library, we have like, you know, the top, the three options and stuff. It, you know, it looks more legible in my opinion.
Yeah. So that's only for practice, not for, yeah. So if you want to do it, like to help you study for the lab, then you can, but it's not required. Yeah. That's the one that you can download, you can print out. It is all of them, but if you go onto Top Hat, it has the individual ones. So you can just download that lab and print out individual labs if you want to. Yeah, it'd be better probably to like have a bookmark for it or something. Because if I set up everybody clicks the wrong thing every time to set up their account, you know what I mean? If you don't have this and this is fine, have you tried to get into Top Hat yet? So, go back to, let's say, assignments, I think. And again, we're gonna sort it by type. Yeah, and if you've done it already that way, when did you sign up? Did it open up and everything? Did you get like access? Then you can just go through like Top Hat's website after that. It's better. Yeah. The only reason I don't put the link on there is because literally everybody will put that link instead of clicking this to access their initial, but you can still get to it. But it's, yeah, everybody's always clicking the wrong link otherwise.
As the Top Hat, so you said I don't have to use my OSU email? That was the one that I was like, it wasn't working for me.
Okay. Sure. And it should be through your OSU email. You should not have to do it this way. And it didn't open up whenever you did it the other way?
No, and then I went to the OT thing and tried to log in the Orange Mail, and then it didn't reset my password, and it's still not working, so I emailed IT, and maybe that's what it was. I was getting excited.
But it's working for you on Canvas. I mean,
Yeah, Canvas is working great.
It's the same sign. I mean, you shouldn't have to sign in to Top Hat. Okay. It should just automatically go if you click on it.
I wish the Cowboy Mail was linked to this one. I guess I'll just see what I can do. My son was, earlier it was on the story, my son had to go. Don't know if that's why.
Yes, you have it.
I can mess with it when I'm trying and stuff.
I can. It's working for other people, but like that doesn't help you. So this shouldn't be what it's doing. And I bet maybe it has to do with your OP stuff, because this isn't. Might email the bookstore. Okay. I don't know why it's not. Yours isn't working and other people's is, and I don't know why. I have no explanation other than if it is an issue with your OP. I don't know about it.
Okay, yeah, I'm not great at technology, so that's why I kind of get anxious about it, because I'm like, okay, thank you.
Yeah, good luck. You can email me if you can't figure it out, and we'll try something else.
Okay, I appreciate it. And all the exams will be in here or in a different one?
In here. For lecture, anyways. Lab will be in lab.
Upload and podcast.
Huh?
The closet for the
Maybe up higher.